Hello, and welcome to Field Lab Earth, the podcast that's all about past and present advances in the fields of agronomic, crop, soil, and environmental sciences. Today, we'll be talking to Dr. Sakiko Okumoto, William Rooney, and Subbarao Guntur. When we fertilize our crops, some of the nitrogen from that fertilizer gets converted into different forms through processes called nitrification and denitrification. When non-plant available forms of nitrogen exit the soil through water or as gas, it's a serious environmental problem. Thankfully, plants have some pretty nifty ways to prevent nitrification, such as biological nitrification inhibition, or BNI, a process that manages the bacteria that cause nitrification. In this episode, Sakiko, Bill, and Subbarao join me to discuss Sorgoleone, a product of sorghum roots that plays a key role in the BNI process. Make sure to listen to the end of our episode for a student spotlight as well. I'm your host, Abby Morrison. Let's talk about science. Hi everyone, today we have Sakiko, Bill, and Subbarao with us. Sakiko is originally from Tokyo and earned her bachelor's and master's at the University of Tokyo. She earned her PhD in Tübingen University in Germany, then had a postdoctoral training at Carnegie Institution of Washington in Stanford, California. Her first faculty position was at Virginia Tech, then she moved to Texas A&M to her current position. Bill earned a bachelor's and master's in agronomy and plant breeding from Texas A&M and a PhD from the University of Minnesota. He's been a sorghum breeder/geneticist at Texas A&M University for the past 30 years. Subbarao is a senior researcher and group leader of BNI research at the Japan International Research Center for Agricultural Sciences, or JIRCAS. He received his PhD in crop physiology and nutrition from the Indian Institute of Technology, and then received postdoctoral training in several institutions, including the University of Saskatchewan in Canada, NASA, and ICRISAT. He joined JIRCAS in 2002 and since then has pioneered a research program on biological nitrification inhibition, which is gaining international attention and momentum. Hello, everybody. Welcome to the show. How are you all doing today?
Well,
pretty good.
Good.
Good. So wonderful to have you. So today we are talking about nitrogen and sorghum and many aspects related to that, if everyone couldn't tell from our bios. So to get us started, let's just launch in with kind of an overview of just the nitrogen issue in sorghum, kind of the biological nitrification inhibition issue that this research is focusing on. And Sorgoleone is, in particular, one of the things we'll be talking about today. So if we could get like a high-level introduction to what that is as well, that would be fantastic.
Yeah, obviously nitrogen is one of the most quantitatively important nutrient that we have to add to the field because the amount of nitrogen you add to the field directly impacts the yield. Without that, you can't really have the commercially competitive yield. So that's a fact as of now, certainly. And doing that obviously is going to come with some amount of environmental cost and loss, okay? Because not all the nitrogen that you add to the field stays in the field. Rather, there's this pathway of loss, okay? So first of all, the ammonium or ammonogenic molecules that you add to the field can be nitrified, okay, by soil microbes, and it turns into ultimately nitrate, which is negatively charged, and that can just run off from the field with the next rainfall. And because you are adding so much nitrogen into the field, in a way you are promoting the population of nitrifiers in the soil. Right, so that's one big issue. So first of all, of course, farmers will lose money because the nitrogen is leaving the field and not getting into the crop. And second of all, that nitrate that's downstream, will contaminate fresh water, will contaminate, gulfs or anything that any body of water that eventually the river is going to dump into. That's an issue. And another thing is that in an anoxic environment, for example, waterlogged environment, those nitrate can be actually then reduced, denitrified, And if it goes all the way to nitrogen gas, it's fine in terms of pollution potential, but it can produce things like nitrous oxide as a byproduct, which is a bad greenhouse gas. So what we are trying to do through this project is to prevent the loss of nitrogen by using the plant's natural ability to prevent the nitrification, the first step of the nitrogen loss. Honestly, this was really conceptualized and pioneered by Dr. Subbarao's lab. So, I'm glad that he is here, and you can probably add more to what I said, Dr. Subbarao, but he's really the pioneer of this area.
Okay, to supplement what Dr. Sakiko has said, the nitrogen fertilizers, when we add to agricultural fields, the nitrogen fertilizers themselves are not harmful to the environment. Mostly they are in ammonium form, which can be used by the crops. Most of the crops can use nitrogen in ammonium form. So the nitrogen fertilizers as such are not harmful to the environment. But when you apply to the soil, as Sakiko-san said, that there is a soil bacteria that converts this nitrogen fertilizers, which are not harmful to the environment or to the plants, into nitrates. It is a kind of soil biological process. The specific bacteria lives in the soil that oxidizes the ammonia into nitrate. But once it is converted into nitrate, as Sakiko mentioned, that it cannot stay in the soil, and it turns into a very harmful nitrogen form to the environment, because it doesn't stay in the soil, it just moves into the water stream and contaminates the water bodies. And also it turns into nitrous oxide under certain conditions by another group of bacteria. So it opens up a pathway for nitrogen leaving from the agricultural fields and converting into harmful nitrogen forms, damaging the environment and the human health. And also it's a huge economic loss to the agriculture. Nearly 70% of the nitrogen fertilizer what we apply in agricultural fields is lost, either through nitrate leaching or through the nitrous oxide formations. So the research what we are involved is to try to use the natural plant ability, natural plant function to control the soil bacteria in such a way that they will not be their function is their biological function is reduced in such a way that the nitrogen fertilizer what we apply in the fields would remain in the fields and will help the plants to grow and yield. Because of that, we don't need to apply so much nitrogen fertilizers to the fields as we are currently applying, because we are controlling this nitrogen loss. So we can cut down the nitrogen fertilizer applications to 50, even 60% from what we are currently applying. So that is the essence of this research we are involved in. So that is where the biological nitrification inhibition in sorghum comes into play. That is how the sorgoleone, which is a major component of exudation coming from the plant roots, how that really controls this nitrifying bacteria when it is released from the plant roots. That is the theme of this research article what Sakiko-san has authored.
Fantastic. So the biological nitrification inhibition is basically the plant's natural processes that can help reduce the fertilizer turning into the nitrates and nitrous oxide. And then the Sorgoleone is what's exuded from the roots as part of this process. Okay. I just wanted to make sure that I had my definitions all lined up in a row moving forward. So obviously y'all have been doing a lot of work on this. Thank you for doing that. And thank you for joining us very early in the morning for some of us. And at the end of the day, for others, I know the timing was a little tricky, but I appreciate that. So you, though, have been collating all this information into this review paper. So there were several areas where you were able to kind of look at how this specific chemical or exudation thing is being used to inhibit the nitrification, I guess. So the first one is chemical properties. So could you just tell us a little bit about what you found out about that?
Well, so we didn't really find out the chemical property. That has been already done by some other groups, but this is a chemical that is pretty much a general offender, I would say. So it's a compound that's hydrophobic. It has a quinone-like head and a long hydrocarbon chain tail. So that tail is what makes this compound oily. So it's a really fun compound to work with. It kind of gets exuded from the root hair, and you can actually see it. How often does that happen, right? It's pretty cool. So anyway, what it does is to inhibit reactions that requires membrane electron transport, seems like. So it is a very strong inhibitor for nitrification, but not only that, it does also inhibit the mitochondrial respiration and photosystem too. So the common denominator there is the membrane electron transport chain, because all of them have quinone-like cofactor that is necessary for the transfer to happen. Okay, so for nitrification, we don't have a concrete evidence that indeed that's what's happening. The enzyme that it is working on, ammonia oxidase, is a very elusive enzyme, and we don't even have the crystal structure. Oh, and I think Dr. Subbaro's team actually has the evidence that it works against another enzyme as well, don't you?
Yes, the two enzymes, just a small correction. It's ammonia monooxidase, AMO. The other enzyme is hydroxyl amino oxidoreductase, HAO. The sorgoleone that is released from the sorghum roots affects or suppresses both the enzymes responsible for nitrification process in the nitrosomonas bacteria. That is the bacteria that lives in the soil, responsible for this nitrification process. So it basically suppresses this nitrifying bacteria. This is the sorgoleone compound that is released from the roots.
Okay. So what does it look like?
Yellowy browny oil.
Actually, you can see these yellow oil droplets coming from the sorghum roots. You can see with your own eyes. When you wash the roots with an organic solvent, the whole liquid turns into yellowish liquid.
And is that just naturally exuding? It's not like a result of like, oh, it's like an injury and now it's gonna exude. It's just like a thing that it does.
They do it naturally.
Amazing.
It comes out from the plant root system. The sorghum roots naturally release this one. So what we are trying to do is we have established that this compound specifically affects this nitrifying bacteria, and it affects in such a way that it reduces the soil nitrate formation in the fields. That is what we have established from the last 10 years of research in JIRCAS and with the partnership with Texas A&M. And having established the primary function of this compound coming from the sorghum roots, the next step is how to improve this plant function in sorghum in such a way that we could develop the sorghum varieties that can produce a lot more than what we normally see in most of the sorghum varieties. That is where I think the sorghum breeders, like Bill, role comes in. How to maximize, how to improve the sorgoleone release using the breeding approaches in such a way that we could develop the sorghum varieties that can produce maybe 5 times, 10 times more than most of the current sorghum varieties produce. That is how we could control this nitrification process in soil. That is how we can reduce the nitrate formation. That is how we can reduce the nitrogen fertilization for sorghum field, for sorghum production. Okay.
Yeah, my role in this is one of how do we go about making this trait better? Do we need to make it better? What's the status? And so some of the answer to that question is twofold. One in that you can look at the underpinnings of the genetics of the trait, and the second is you can simply look at material and find out what the status of material is from that standpoint. Subbarao has found some exotic accessions in his early work that are, well, one in particular that's extremely high in Sorgoleone production. From a practical standpoint for breeding in the U.S., however, that accession is a lot of work to work into a breeding program. And so one of the things that we did is to just evaluate germplasm in our own program and find out what kind of range of variation we have. Is there anything of value there? And what kind of background is it in? Meaning, if it's in a high performing genotype already, that means we have the ability to distribute something and make something available quicker and discriminate between things. So I should say, Sakiko's group did the phenotyping of this trait, because that's not easy to do, and it's not something that we can do in the field on any scale. So she did lab assays to score for Sorgoleone concentration in a series of about initially 100 different things, looking at range. We found good range, and so then we evaluated another set of about 200 materials looking at the range and variation of that. Those 200 materials were mostly hybrids that we've developed in our program, that we have already vetted agronomically for performance in grain sorghum production regions of the United States. And we were able to find some things that, although not as high as the standard check that Dr. Subbarao uses, they in some cases are not statistically different from that, and we see those same trends manifest in small plot trials where we can actually go out and measure N2O emissions and look at the BNI rates. And so in doing so, we've been able to find things that are pretty good already, all right, and work with those. In conjunction with that, we're going back and looking at the genetic inheritance of the trait, building or looking for QTL and loci in the genome that are consistently associated with this. That's what Sakiko and I are spending a fair amount of time working with some populations. And then finally, because this trait is not easy to phenotype, building prediction models so that we can use genomic prediction and tools like that to actually do the pre-breeding once we've got this. If we've got this in the system, use that to pre-breed and then pare down the amount of material we have to phenotype to select for further improvements in the future.
You mentioned that it's like a difficult thing to phenotype, which is interesting because you can kind of see it. You can change the color of the water. I mean, I know that's not like exact to just be like it's more yellow or whatever. But is it just because it's on the roots where it's not something where you have to like dig it up. You can't just look at it while it's just growing naturally. So I'm kind of curious what difficulties there are in phenotyping and a little bit more about kind of how that process works.
I'm going to let Sakiko give you the details of phenotyping. Suffice it from a breeding perspective, her lab did those 200 samples that I talked about in replicate, and that took us, 6 months?
Something Sakiko? Like that, yeah.
Okay, yeah. There's your challenge. It just takes a long time, all right? And that's not, 200 just sounds like a fairly good number, but for a plant breeding program, 200 is not that much. So you want to give her the details, Sakiko, on the methodology?
Yeah. So yes, we can, see the droplets, but that's not enough, right? So to actually quantify it, we have to grow the plants in the lab and set condition, extract the root with the organic solvent, just like Dr. Subbarao alluded. But then that has to be shut down the HPLC to quantify the compound. Okay. And then that has to be normalized against the dry weight of the root, which means that you have to get the dry weight of the root. So, it's not conceptually difficult, but it still takes time. And, if you think about other like agronomical traits that you will, phenotype in the field, like Bill is always, getting the yield data, plant height data, maturity, etc. Those are done in thousands. Okay, so that's what Bill is talking about. It's not something that we can scale very easily. So that's why the genomic prediction is important, because we have to. It's very hard to do this in thousands.
Yeah. Is this something that the plants are exuding from the get-go, or is there a level of maturity that they have to reach before they'll really start cooking on that?
Okay, so that's a great question. We actually have a separate paper on this, but from the plants growth perspective, yes, they are pretty much secreting from the get-go. We can see the secretion after 3 days.
Oh wow! Okay.
After 3 days after germination, yeah. Now, if you look at a very detailed developmental stage of the root, very youngest of the root, the root tip zone doesn't secrete this compound, but that's another story from the, like a scale of the growth season. I think we can say that they're secreting it from the get-go. And I think Dr. Subbarao already published the data a while ago showing that the secretion is more or less stable throughout their life cycle.
It increases with the growth as the plants grow, but usually it reaches the peak around the heading phase. And then once the plants enters into the active grain filling period, then I think it stops. Up till that point, it grows in a more linear and logarithmic fashion. But during the grain filling process, it completely stops. Most of the BNI production, including the Sorgoleone, coming from the root systems.
That makes sense. Yeah, I mean, if you're trying to keep that fertilizer in the soil, then at a certain point, you're like, I'm good. I got what I needed. We have talked about some of the chemical properties. We've talked about kind of having that range of kind of naturally occurring or how often it occurs in these hybrids. But some other areas in your review that I'm hoping we can touch on, we've probably touched lightly on many of these things already. But the kind of biological activities and biosynthetic pathways. I know, Bill, you kind of mentioned a little bit about heritability, but if there's anything you want to add on that. And then also just its utility in nitrogen use efficiency would be fantastic. So any of those topics I would love to explore.
So when you look at this trait, and I'm just working off of my experiences and what I've read from others in sorghum, and you compare it to other crops. But one of the things that's interesting about sorghum is that we see an incredible range of variation in the Sorgoleone production. And there doesn't seem to be a pattern as to why certain genotypes have it more than others. And I'm speaking primarily based on our screening material. Our material is representative of the germplasm that's in the U.S. production system, and we focus primarily on grain, but we've looked at some forages and we've looked at some bioenergy things as well, and they all seem to have it. They all seem to, and then there'll be some genotypes that don't have it. Okay, so or have much lower concentrations of it relative to those. What that told me is, as we started this project, is that we haven't been selecting for Sorgoleone and BNI production, but we haven't been selecting against it either. All right, so it's just sort of been floating around in there. The crops probably always had it. You can see the ecological advantages to the trait. And so whether or not it was maintained really became just a product of chance, for lack of a better term. So when we looked at the heritability of this in the material we tested, and this is based off of that 200 genotypes that we screened in the last set, because we had the right structure of family relationships to look at that. The heritability is pretty high. On a quantitative genetics estimate scale, we're looking at about 0.6 to 0.65, which would definitely put it in the high category. So it is selectable. I think that we will have the ability, once we get started with prediction modeling, to screen lots of material, and if we can use the prediction modeling to get that material pared down to a manageable number for actual phenotyping, we're going to obviously want to see if our prediction modeling is working by finding the best things. And if it does, it gives us a clear path to maintaining our numbers in breeding, and then also working with phenotyping at a reasonable scale. Now, the weird thing, or not the weird thing, but the interesting thing about this is that you have a pathway that's pretty well defined, and don't ask me to tell you about it because I don't remember those things, but these other folks can do that. And so it implies that this should be a even higher more heritable thing. If I can see the pathway, well, if I knock out those genes, and they know that because Sakiko is working with some knockout mutants that basically don't produce anything, right, Sakiko?
Yeah, so there is a RNAi knockdown line that's already available from a person who basically worked out this biosynthetic pathway, and that one is pretty much null. It doesn't secrete anything.
Yeah, so you have this that sort of further supplements it, and so a logical question is, well, why isn't the heritability 1? And the heritability is not 1 because there's a lot of other factors just besides that biochemical pathway that are causing the concentrations that we're seeing. I mean, one of the first things that became obvious to me, and we're still working through that, is association with seed size, okay, and how that plays into these types of things, and what the consequences of that are. But there's other environmental factors that are certainly affecting it as well, and many of those we still don't know, and so that's going to take some time to figure out if we have the time, effort, and money. On the other hand, as a breeder, sometimes I don't worry too much about why something works. I worry more about whether or not I can capture it and I can use it.
Hi everyone, I hope you're enjoying the show. Interested in learning more? Sakiko, Bill, and Subbarao's paper, Synthesis, Function, and Genetic Variation of Sorgoleone, the major biological nitrification inhibitor in sorghum, published in Crop Science, is always freely available. If you are a certified crop advisor or certified professional soil scientist, you can take a quiz for continuing education units for this episode, which can be found in our show notes or on certifiedcropadvisor.org. Let's get back to the show. Working through our lovely list of options, I think biological activities might still be on the list, and then the utility in nitrogen use efficiency overall.
Yeah, I think we have established from the last several years of field trials and several other experiments that the amount of the sorgoleone coming from the root systems largely determines the inhibitory power, the inhibitory capacity. The more sorgoleone comes out, more the bacterial suppression, but the more suppression of this nitrifying bacteria you can see in the field. That suppresses the nitrate formation. So that really improves the nitrogen use efficiency because it reduces the nitrate formation, nitrate loss. both under low nitrogen input conditions, like in African conditions where there's not so much nitrogen fertilizer is applied, but still nitrogen, plants need to take it up. That nitrogen normally comes from the soil organic matter mineralization, which still has to go through the same. It releases the ammonium, and which again are subjected to the nitrification processes. So even if you don't apply nitrogen fertilizer, nitrogen still can be lost through the nitrate leaching. So the Sorgoleone production under low nitrogen availability is much higher than when you fully fertilize the fields. So in some ways, the high Sorgoleone production actually improves the productivity under low nitrogen input conditions, systems like what you often see in Africa. So this is very important. The high Sorgoleone production, high Sorgoleone producing varieties would actually improve the adaptation to many African production systems uh, where sorghum can be grown. Yeah.
So there's always the risk of like too much of a good thing, or we talk about on the show how, certain like at a certain point adding more fertilizer is like not going to help you. Is there any kind of sense with Sorgoleone at this point of like a certain point where it is just not helpful to exceed that amount, or potentially is like harmful if it's suppressing too much bacteria or like other kinds of bacteria? Is there any indication of that, or are we still kind of too early days to know about that?
There's always a possibility, okay, because like I said, this compound is a general offender. That being said, Bill's materials that we screened are already vetted for the yield, all right? And then one of the first things, honestly, what we did was to, once we get the ranking of Sorgoleone secretion, we tried to see if there's any correlation with yield. The number one thing that you care about, especially if you're a breeder, is that is there any yield penalty, right? There was none. Okay, so up to that level that we saw in the pool, we really didn't see. And the wild line that we always use as a check that's discovered by Dr. Subbarao is, they're really good at putting on the biomass, okay? And the impact on the soil microbiome is something that we want to dig into. Dr. Subarao's team did some of the shotgun sequences, and there is change in soil microbiome, but is that a functional change, or is that going to affect the nitrogen cycle, carbon cycle in any harmful way? That's not discovered yet, so that's something for the future research.
I think we have done a lot of work in Brachyara systems where the BNI function is one of the strongest. And also recently we have been looking at the BNI wheat systems to see whether there is any significant changes happening in the soil microbial population. We don't see any major shifts happening. The only, because the BNI compounds coming from the root systems, whether it is Sorgoleone or something other compounds, their main function is only on nitrifying bacteria in the soil. They don't seem to affect the bacterial activity. In fact, actually, some of the BNI compounds stimulate the mycorrhizal association and other beneficial organisms in the soil. So in such a way that it actually benefits the overall the microbial activity in the soil. So we have not seen so far any negative impact on the microbial activity in the soil apart from suppressing the nitrifying bacteria.
Fantastic.
I think I'm going to add one thing to this, and Sakiko, you can tell me if I'm stepping wrong. We have a wild relative of grain sorghum and forage sorghum called Johnson grass. All right. And Johnson grass is really good at using this trait to scour, as you might expect it would be. All right. And if those numbers are any indication, we can do a lot more in terms of increasing based on simply what's there.
Recently, we identified a new Sorgoleone analog that seems to be produced a lot more at higher levels in Johnson grass compared to many of the cultivated sorghums. And this new analog seems to be much more stronger inhibitory function than the normal Sorgoleone what we see, maybe 2 times more powerful. That is detected in the Johnson grass. So probably we will have to really look at it a lot more accessions to see what is so special about the Johnson grass, whether any new types of Sorgoleone analogs are coming in the Johnson grass compared to the cultivated sorghums. That remains to be, I think, still it's unknown, but remains to be discovered.
Wow, that's amazing. So I think we've covered most of the larger kind of areas that were in your review paper as far as kind of where we are now, what our current understanding is. But we've already mentioned several of them, but there are many areas in this kind of new area that we are exploring to go after and study. So where is the future research headed for Sorgoleone?
Yeah. So multiple things. So just Bill already covered quite well what we can do with breeding, and I think there's a lot of gain to be had from that. Another thing that I'm also interested in is to dig deeper into beyond the genetic mechanisms beyond the just biosynthetic pathway. Okay. So is there some regulator, for example, that would control the whole, set of genes? And those are usually a much better target compared to the, each individual downstream enzymes. So to discover those, and that's one of the things that we would like to do. Another thing is that we have to make sure that this matters in the field, right? So like Dr. Subbarao mentioned, he has done some pioneering work showing that yes, BNI actually increases the nitrogen use efficiency, but that has to be tested in the context of agronomical practice as well. So, for example, what we do in the United States with the hybrids and high yielding variety is very different from what might be done in sub-Saharan Africa, right? So here we are trying to trying to prevent the loss of excessive nitrogen fertilizer. There you might be trying to like keep what precious little nitrogen you have in a rootstone, right? So we have to be able to evaluate the benefit from the agronomical systems. So that's why we have Nithya Rajan. She's an agronomist, and she's also capable of doing like the greenhouse gas analysis, etc. So she's looking at the system level nitrogen economy and the nitrogen use efficiency, and that's very important to do because after all these work, if the gain is by 5%, no one's going to care about it. But because Bill already has a agronomically competitive lines that has substantial BNI, we can first use those to prove the concept. Okay, if we have, let's say, 50% decrease in Sorgoleone, we see this result, right? Can we reasonably expect to reach the goal of, let's say, cutting nitrogen fertilizer by 50%? Ultimately, that might be something that we aim at, like just Dr. Subbarao said. So, yeah, so it has to be the multidisciplinary thing where we work together, and what we have from the breeding side is fed to the agronomy studies and validate the value. So, and that takes time.
You also, I'm going to throw this out there too for the audience in general, that these things they occur in an economy, and so we've got a trait here that we think is valuable, but that has to be understood as having value to the people that will benefit from that. And this is a hard one because you're asking people to believe that there's something in this plant that's going into the soil and it's reducing your need for additional nitrogen. So you're going to have to prove that that's actually the case before people will invest extra money to recoup that value, because it's not like herbicide resistance, which you can just spray and see. And then the second thing is that there's a component of this from the climate standpoint. We're reducing into emissions, and that should have value in terms of greenhouse gas emissions. But that is a system that really still hasn't been quantified into how you capitalize value to bring and incentivize people to actually adopt and use these things. So there's a lot of things that have to be done at the system level and at the incentive level to demonstrate to producers that, yeah, we have things that are better than others. Are they worth the extra cost or value that they may have to them to use those? And this is just like any new trait. You've got to figure out a way to do that. And some traits are easier than others. This one's going to be an interesting exercise in doing so.
Yeah, totally. Is there any kind of legacy effect that Sorgoleone can have? Does it build in the soil, or is it just kind of like it's just right by that plant and the plant stops making it and it like breaks down and goes away?
I think what we have seen is only about maybe 2 to 3 months maximum. The impact will be there. After that, after 2 to 3 months, we are not so sure how long that effect lasts. So it definitely helps the sorghum, the crop to utilize the nitrogen that is applied. But subsequently, how that affects the following crop, that remains to be seen. We still don't have full understanding on that.
Cool. Yeah. And I was thinking, oh, it'd be cool if it could help with like legacy end, but then I realized it's trying to stop it from like leaving the system. So that wouldn't really work. Okay. So I have been talking to you guys for quite a while, which has been lovely, but I don't want to keep you from the rest of your days too much. So I have 3 questions left for you. The first one is, if listeners want to learn more about this, where can they go to learn more?
Yeah, there are several reviews coming from our groups and several other groups coming published on giving the current status of this research. So people can easily look into it to get more information, or maybe contact some of us if somebody wants to know more about it. The JIRCAS organizes a consortium, BNI consortium meeting every 2 years, bring together some of the researchers working on this with us and other groups. That is one forum. If somebody wants to know more about this research and wants to have a contact with some of us attending the BNI consortium, we organize every 2 years. That is one forum, and some of the articles coming out from the groups and the reviews.
Great. So we've mentioned a little bit of the things that people can do to get involved, but what can people do to get involved with this kind of stuff?
If you're talking at the farm level, I don't think we're at that point quite yet. We're still got to get some documentation to justify why this is going to be valuable to a producer and what's going to draw them to it, or what's going to draw someone who's interested in carbon credits to it. And that's something that we have discussed quite a bit internally as to how do you go about doing that. It's going to require more work. And so I think that just awareness of it and people being aware of it would be helpful, just that they put it on their radar as something that could help in the future with nitrogen use and nitrogen applications. And then ask about it so that it's at the forefront of people that are influencing how those decisions are made to generate funding, to do the testing and science that's required to make this come to fruition.
I think the immediate impact I can see is reducing substantially the amount of nitrogen fertilizer applications for sorghum production system. I think that is going to be the immediate impact. Once we are successful in developing high Sorgoleone producing hybrids that are commercially available, that can cut down the nitrogen fertilizer applications to anywhere between 30 to 50%, based on what our understanding. I think that is going to be the immediate benefit for the farmers. Once the BNI sorghum varieties hybrids comes into the market.
Perfect. Yeah, wonderful. Keep an eye out for that if you are a farmer, and keep an eye out for maybe some of those conferences if you are on the research side. So then final question for you. What is one fun fact about you that listeners wouldn't know if all they had was your research?
I'm a runner. I do running for hobby, and I have my first 50K race coming up.
Wow. That's very impressive.
Yeah, this is a stupid idea. I'm turning 50 in just a couple of weeks. 50 for 50.
There you go.
Wish me luck.
There's really nothing too interesting about me.
Oh, come on.
I run cows. I have about 25 Black Angus cows. So that's my manual labor job.
Wow. All right.
Okay. Well, if I can sub anything for Bill, there's no question in the domain of agriculture that I asked Bill that he didn't have an answer to. He is extremely knowledgeable.
Well, when you've been steeped in it your whole life, that helps.
Yeah.
So.
I enjoy what I do. I guess that is my hobby. So doing whatever I am doing is my hobby. So I think that's what keeps me going, I guess, all these years.
That's wonderful. That's a good attitude to have, of enjoying everything you do to the point it could be a hobby. That's wonderful. What a positive outlook, and what a positive way to end this conversation. This has been wonderful. I have learned a bunch more with all of my bonus questions, and y'all have been very good and patient with the many bonus questions I had. So thank you so much for your time, for all the research you're doing. It's been a pleasure and an honor to have 3 experts in this call. So thank you for everything you do.
Thank you.
Thank you.
Thank you.
Hi, everyone, and welcome to our Student Spotlight, where we highlight the work of graduate and undergraduate society student members. Today, we'll be talking to Megan. Welcome to the show. Can you start us off by introducing yourself and where you're studying?
Hi, my name is Megan Baker. I grew up on my family's farm and ranch in eastern Nevada, and I'm currently pursuing a PhD in plant science at Utah State University, which is also where I did my bachelor's and master's degrees.
Great. And what are you currently researching?
So my research is, I'm on a couple different projects, and they're all mainly focused on nutrient management. And so I'm looking at updating different phosphorus, potassium, zinc, and sulfur fertilizer recommendations, because most of our recommendations are a couple decades old, and we want to make sure that USU's recommendations for fertilizer are up to date.
Wonderful. And if you could have your dream research project, what would that look like?
I think it'd be really cool to do a project where you kind of compare, whether it be soil sampling, forage sampling, or any of that, methods that actual farmers and agronomists use, and people in the industry, to like how we do in research, and see how much of a difference there is in the reported results, and see if we can either make it so when we're giving recommendations or methods that they're more tailored to either how they do it, or find a way to make people kind of update what they do so that they're getting accurate results, so better better results out of what they're putting into it.
Wonderful. Well, if you'd like to get in touch with Megan about her work, we'll have her contact information in our show notes. Thank you so much for being on the show today, and best wishes on your future studies and career. Thank you.
Thank you.
Thank you for listening to Field Lab Earth. More information can be found in the description below. Thank you.